Hello and welcome to your video lecture on section 15.3 on eukaryotic transcription. So most of these processes are going to have three phases. So we've got, or you can even think of them as steps, three steps. We've got initiation, elongation, and termination. And our learning objectives for this section is to list the steps of eukaryotic transcription, discuss the role of RNA polymerases in transcription, and compare and contrast the three RNA polymerases and explain the significance of transcription factors. So we'll start with that first step, which is initiation of transcription in our eukaryotes. And we are going to discuss our three polymerases here as well. Our eukaryotes are going to use three different polymerases. We've got RNA polymerase 1, 2, and 3. We use these Roman numerals to refer to them. And we also have a distinct set of transcription factors to help bring it to the DNA template. And we're going to talk more about these factors on the next slide. So our transcription process is said to be monocistronic. It means that each is going to transcribe a different subset of genes, creating just a single polypeptide, whereas in prokaryotes, we're able to do multiple ones at the same time. So we're going to call the prokaryotic transcription more of a polycistronic system. So our pre-mRNAs are then going to undergo an extensive processing after transcription to go into the process of translation. But let's first talk about our three different types of RNA polymerases. And so we'll start with the first one naturally. This is going to be found in the area of the nucleolus. So remember that we find the nucleolus within the nucleus, and this is going to contain a nuclear substructure where our ribosomal RNA is going to be transcribed, processed, and assembled into ribosomes. So we're going to talk more about this, a ribosomal RNA, toward the last section of this chapter. But essentially know that our RNA is a component of the ribosome, and we absolutely need it for our last process of translation to take place. Our next RNA polymerase is going to be the second one, and this is found within the nucleus. And this is really responsible for transcribing the majority of our eukaryotic genes. So when we talk through the process of transcription, this is the specific type that will be the most functional within it. And then we've got our third type, which is also found within the nucleus, and this is going to transcribe a variety of structural RNAs. So a lot of our pre-rRNA and our transfer pre-RNAs and our small nuclear pre-RNAs. We're going to define all of these structures in one of the last sections in this chapter. Now you may be wondering why I haven't mentioned this column just yet. This column is referring to a oligopeptide toxin that is produced by the fly agaric toadstool mushroom and other species of the Emini genera. And this mushroom was actually used to kill flies. They used to take the mushroom and crush it up, and I believe put it in some milk so that flies would be attracted to it. And then, of course, as they went into it, the toxin would kill them. So what scientists have found is that they can characterize a new gene by figuring out which polymerase transcribed it by testing whether it is sensitive to this alpha amanitin. And what they found is that polymerase 1 is insensitive to it, meaning the polymerase can transcribe the DNA in vitro, meaning in the body, in the presence of the toxin, whereas type 2, the toxin prevents the enzyme from progressing down the DNA. So it's really inhibiting the transcription because this is very sensitive to it. And then we have our third type in which it is moderately sensitive to it. So as we said in the previous slide, RNA polymerase II is going to be the main one to transcribe our mRNA. And so what we need to do is look for a promoter for the RNA polymerase to be able to find the correct place to kind of latch on to the mRNA strand. And this is known as the TATA box. And it has consensus sequence of TATAAA, although there are some other promoters that include a CAAT box, a GC rich box, which means we've got a lot of those nucleotides in a row, and then an octamer box as well. Now you may be wondering, why did the body choose this TATA box to be the promoter? And the explanation is that a lot of the AT bonds have a low thermostability, and so this really helps the DNA to easily unwind to prepare for the process of transcription. Now we also need some help of some basal transcription factors, and these are going to bind to the promoter area as well, and it kind of waves a flag to say, hey, RNA polymerase II, come latch on over here. And we also have these other specific transcription factors. We've got 2A and 2B, and we can continue adding more of them that were named basically after the letters in the alphabet, which can span from A to J. But the core complex is going to include our transcription factor 2D and that TATA binding protein. And by having all of these factors and proteins in place where the promoter is, it helps to stabilize this pre-initiation complex and recruits RNA polymerase to attach. Now, sometimes we have enhancers and silencers, and they can affect the efficiency of transcription. So we don't necessarily need them for transcription to continue though. Now, if we're going to utilize RNA polymerase I to transcribe our genes, we are going to have 2 GC-rich promoter sequences. And what was found is that when we have additional sequences that we find further upstream from that initiation site, it can help to enhance initiation. So for instance, we saw that our TATA box was over here, but if I looked further down the line and saw that we had some other promoter sequences, such as a CAAT box or a GC rich box, then that's going to help enhance initiation. And our genes that are transcribed by RNA polymerase III have upstream promoters that can occur within the genes themselves as well. All right, now we've moved past initiation, and we're going to talk about elongation and termination in transcription. So our polymerase is going to be released from the transcription factors, and we are able to move that RNA polymerase down the mRNA strand, and it begins to synthesize the pre-mRNA in the 5' to 3' end. Now again, if we remember what DNA looks like, we know that it is a double helix. So we are going to need to separate that helix in order to read the one template strand. And so we're going to utilize FACT, or what's known as facilitates chromatin transcription. Essentially, it is a special protein complex that's going to help to pull the histones away from the DNA template as that polymerase moves across. And as we are creating our mRNA strand, we are going to have our FACT complex replace the histones to recreate the nucleosomes that once were. And then once we head down the end, our pre-mRNA tail is going to be removed by cleavage during the mRNA processing phase that we're going to talk about in the next section. And our two polymerases, polymerase 1 and 3, are going to be required to give termination signals.